For this introduction, I am very happy to have the opportunity to introduce our company, Navignostics, to you and to explain how we help find the right drug for each individual cancer patient. A little bit on our background first. So we're a spin-off company from the University of Zurich, specifically from the Bodenmiller lab. And the Bodenmiller lab pioneered spatial tissue analysis of tumors, and for over 10 years we showed repeatedly how powerful our approaches are at characterizing tumors, at understanding how different patients react differently to different treatments, have different clinical outcomes. And so with Navignostics, we just wanted to push this one step further to actually directly benefit patients by developing novel diagnostic approaches to inform the best treatment decision for each one of them. And that's what we did. So we established by now a team of 15 people in Switzerland, in the Zurich area. And it's a fantastic team that is now working on our diagnostic developments and is already engaged in more than 10 collaboration projects that we have with our clinical and pharma partners. But to take one step back, why did we actually want to start Navignostics? So we saw that there's lots of already available targeted cancer treatments approved now, and many more are being developed. So the amount of available therapies is growing rapidly, but unfortunately that isn't directly translating into the patient benefit because it just shifts the problem to the clinicians. Because the clinicians now have to decide which one of these drugs or even which combination will optimally benefit each individual cancer patient, which is not an easy task because essentially every tumor can be unique and can have a unique combination of targetable features. So this is where we come in with Navignostics, because we analyze the tumors from the patients who need a treatment decision, extract this molecular tumor data that is highly relevant to make an informed treatment decision, compile it into a report that we return back to the clinician. But importantly, we also store this molecular data that we extract together with the information how the patient then responded to the treatment that they received. And this allows us to build a very valuable database over time that will allow us to continuously identify new biomarkers and improve our treatment predictions. So now you're probably wondering what these molecular data are that we are extracting from these tumors and how they're different from existing diagnostic approaches. So what we extract from the tumors is spatial single-cell proteomic data. In a way, it's pretty similar to what they do in routine clinical pathology in that we measure proteins in tissues. But in routine pathology, using IHC typically, they can only measure one protein in a tissue section at a time. And this is pretty limiting in regards to the total amount of potential drug targets that you can test. And it takes a lot of tumor material, which is sometimes scarce when you only have finable biopsy material available. On the other hand, we also have genomic analysis of tumors, which was a great step forward in the field of precision oncology. But unfortunately, genomic analysis only gives you an indirect information about the actual drug targets, which typically are the proteins. So this is why we bring our spatial single-cell proteomic approach to the table, because like cancer genomics, we can simultaneously measure many different markers, but directly on the level of the protein, so the actual drug targets, as well as the protein modifications, giving us information about the activation of signaling pathways. And all of that we quantify on the level of the single cells. So we can identify different types of tumor cells and immune cells that are present in the tumor microenvironment. And we can see where those cells are, how they're interacting with each other, whether the immune cells are infiltrating the tumor, which particularly is very relevant information for immunotherapy treatment decisions. And the technology that allows us to extract this data from the tumors is called Imaging Mass Cytometry, an instrument provided by Standard BioTools. And again, it works pretty similar to routine clinical pathology immunohistochemistry approaches because we stain proteins with antibodies, but instead of labeling the protein with a dye, we label the proteins with metal isotopes of known atomic weight, and therefore we can simultaneously stain the tissue with 40 to 50 different markers at the same time in a single tissue section. And then we go in a single measurement step. We measure all of those metal isotopes. We do that by laser ablating the tissue spot by spot, 1 micrometer at a time, 800 laser shots per second. And then the metals in each ablated spot are measured by a time-of-flight mass cytometry system. And each of these ablated spots then becomes a pixel in the digital image that is reconstructed of the tissue. And the different proteins that were measured are represented as the different color channels of this image. And the really most important thing for us, why we use this technology for this clinical approach, is that it's perfectly integratable into the clinical routine, because it works on routine FFPE tissue sections on routine pathology slides, which are very easy also to handle logistically and can be sent by a mail courier, for example. But there are many other reasons why this technology is the technology of choice for us to bring into this clinical space. One of the reasons is that it's a more matured technology by now, so there are lots of peer-reviewed publications, not just from us, but also from many other research groups, over 150 by now. And at the same time, it's also already featured in about 15 clinical trials. In addition, thanks to the labeling of the antibodies with a metal isotope, we don't suffer from any of the typical fluorescence-related artifacts. So no autofluorescence of the tissue, no spectral overlap. And it is also a technology that's very well suited to the analysis of small amounts of tumor material. So we don't need a lot of tissue. Fine needle biopsies are perfectly fine. In addition, due to that we only have a single measurement, staining and measurement step, we don't have to cycle, so we also don't suffer from any of the artifacts that will come from tissue degradation from repeated staining cycles. And we have shown that this technology can be used very efficiently and very fast, so we have demonstrated that we can return a report in under 72 hours from when we receive a patient sample. And what we have really done at Navignostics is we've optimized and standardized all of the workflows around this technology. So we standardized and, to a large extent, automated the entire experimental workflow, and also, of course, particularly important, the data analysis and the reporting. And we have really optimized this for this routine clinical use, as I said, to be able to return a sample in under 72 hours because there is a patient waiting for a response. Another important part of our developments are the panel development, of course. And we're developing these ready-to-use modular panels that can also be further customized with specific targets of interest. Our typical panels are composed of these few modules. We always have the tissue architecture module, just informing on the rough different cell types, tissue structure that is present. Then we have a drug-informing module that informs on the different drug targets on the tumor cells and the signaling pathway activation. Then we have an immune-informing module that in detail characterizes the different immune cell types that are present, as well as immune evasion mechanisms, anything that is relevant for immunotherapy treatment. And then the last module is the tumor-specific module. And all other modules are essentially tumor type agnostic. So we can transfer them from one tumor type to the other relatively easily. And the only module that we have to exchange is the tumor type specific module, where we have the typical markers that are used also in pathology for those respective tumors. And there we now have panels available for colorectal cancer, lung cancer, melanoma, and also breast cancer. And this is an example of what this then looks like. This is a melanoma tumor. So here I'm just showing you a couple of example proteins highlighted in colors. Of course, these are pseudo-colors because what we actually measure are the metals. And we could visualize these proteins in any colors and combinations we would like to. And here in light blue, you can see the melanoma cells in this melanoma tumor. And in red, you can see the CD8 positive T cells. And in yellow, the myeloid cells. And then you can also see a hypoxic area of the tissue here in green. So already from this image, you can get a visual impression that there's a direct contact between the tumor cells and the immune cells. The immune cells are infiltrating this tumor. And when we look at the right-hand side here, this is the exact same tissue area, just with different proteins highlighted now. And now we can see here in yellow the expression of PD-L1 on the tumor cells. and how it is co-localized with the red expression of PD-1 on the immune cells in this MHC class 1 expressing tumor. So already from this, we can get an impression that this patient might be a pretty good candidate for immune checkpoint inhibitor therapy. But of course, we don't usually analyze these images by eye. It's always nice to be able to look at them by eye also to confirm what you quantify, but all that we do, we quantify with our computational algorithms that work fully automated. So from the moment measurement is complete, our pipeline detects the completed measurement and sets off the processing pipeline. First imaging processing, then single cell identification. So we segment the single cells. We quantify their protein expression levels, all the different single cell spatial features. And then we quantify the different tissue compositions, the ratios of different cell types, the cellular organization, so infiltration score, interaction score between different cell types. And all of this we then compile into a PDF report that we return back to the clinician. In the past, we already had the chance to demonstrate the usefulness of this data that we return to the clinician. We were part of this observational clinical trial called the Tumor Profiler Study. And this, what I'm showing you here, is a case example from this trial. So this was a stage 4 cutaneous melanoma patient, 55 years old, for whom all available treatments had already failed. And at this point, the expected survival time was 3 months. Then we received a sample from this patient, and keep in mind, routine pathology and genomic analysis could not come up with any further treatment option for this patient at this stage. When we analyzed the sample, we showed that there was a high overexpression of protein called c-Met, for which targeted treatment was available. And in addition, based on the location of the immune cells and their phenotype, we found that in combination with this targeted therapy, the patient might well benefit from an immune checkpoint inhibitor therapy as well. And this patient was actually treated with this combination of drugs that we recommended and survived for 17 months instead of the expected 3 months, which at this stage in the disease is a very, very good outcome. And actually, just recently, I heard from our clinical collaborators on this trial from another patient that received, based on the data we provided, an experimental triple immune checkpoint inhibitor therapy. And it is now 3 years later, and this patient is still alive. But these weren't isolated cases. So this is going to be published soon. It's going to be a cohort analysis on this trial. And throughout the trial, the clinicians found that while this is only an observational trial, they compared it to what they would have expected in similar cohorts. And so when they looked at the beyond standard of care patients, they found that the patients in this trial had an overall response rate that was almost 3 times what they would have expected in a similar cohort. And for the within standard of care patients, they noticed a lower amount of side effects in this trial. And this is now what we, with Navignostics, are pushing forward into the routine clinical use to benefit patients more broadly with this. And we're doing that in a first step in Switzerland. So our first IVD test will be approved and released to the Swiss market next year. But in parallel, we're already now fulfilling all the US requirements as well. So our direct next step, and which is what we're planning right now, will be to roll out this diagnostic test also to the US market. While we're still in the process of getting the certifications that are necessary, however, we can already provide the data that we have to clinicians as part of clinical trials, and they can already consider them together with the other information that they have available. And that's what we're doing as part of this clinical trial that we have ongoing right now, together with the University Hospital of Zurich. And this is a lung cancer trial, and these patients need treatment decisions, and these are patients that have progressed into the molecular tumor boards. And so then we will receive a tumor sample from this patient and return our spatial single-cell proteomic data in form of our report that I just described, and the clinicians will use that report as part of the molecular tumor board and consider it together with NGS data that they also have available and routine pathology data. Another also very exciting trial we're right now working on is together with a big pathology center in Germany. And this is a melanoma trial to identify the responders to immunotherapy in melanoma. And of course, the aim is, with us taking part in the trial, that we identify a signature that identifies those responders. And as part of this trial, we can directly compare it to the other methods that they're using as part of this trial to analyze tumors. So we can then directly show how much more predictive, how much better our signature is than, let's say, an existing immunohistochemistry approach. These were just two examples of some of the exciting projects that we have ongoing at the moment in Europe. We have lots of different projects with clinicians, both from the pathology as well as the oncology side. And we're also collaborating with pharma companies that are in drug development, where we can also support their drug development process by characterizing the tumors, identifying potential responders, improve the success rate for clinical trials. But what we do not yet have is partnerships in the US, and that's exactly what we're aiming to find now, because our next important step for us is clearly the expansion to the US market, and we would love to already now be able to provide our data to some clinical partners to convince them of how amazing this data is. And so if you or anybody you know might be interested in this, please reach out to me via email or LinkedIn, or stick around after this talk. I will be here for a while, so I would love to discuss further ideas. Thank you very much.